Hello, my name is Patrick Blevins, and today we'll be presenting on MHC types. We'll be comparing and contrasting those. This is for exam 2C. I'm going to give an overview of MHC types. Then we're going to talk about some of the ways they're alike, and then we're going to talk about ways they're different. So let's get started. Uh, MHC, or major histocompatibility complex, is defined as a group of genes encoding cell surface molecules that are required for antigen presentation to T cells and for rapid graft rejection. It's called the H2 complex in the mouse, and in the human, it's called the HLA complex. MHC is a tightly linked cluster of genes present in every mammalian species. MHC got its name from the fact that genes in this region encode proteins that determine whether a tissue transplanted between two individuals will be accepted or rejected. So there's two main classes of MHC molecules, class 1 and class 2. The two molecules are very similar in their final quaternary structure, although they do differ in how they create these shapes via primary through quaternary protein arrangements. Some of the roles that they play, the development of humoral or cell-mediated immune response is one role that they play. They also help with antigen recognition by T cells, because most T cells recognize antigen only when it is combined with MHC molecules. And then they also determine whether transplanted tissue will be histocompatible or histoincompatible. So a class 1 MHC gene encodes glycoproteins expressed on the surface of nearly all nucleated cells. Its major function of the class 1 gene product is presentation of peptide antigens in the TC cells, or cytotoxic cells. The class 2 MHC genes encode glycoproteins expressed primarily on APCs, or antigen present cells, where they present processed antigenic peptides to the TH cells, or the helper cells. So now we're going to talk about how they are like. MHC class 1 and 2 are types of MHC molecules that are encoded by gene clusters of MHC. Both MHC class 1 and 2 are surface antigens that are expressed on cell membranes. Both MHC class 1 and 2 present antigens to T cells. Both MHC class 1 and 2 molecules are involved in the development of immune responses against foreign antigens, and they both MHC class 1 and 2 are responsible for graft rejection during various organ and tissue transplantation. So those are some ways that they are alike. And now we're going to talk about some major differences. I've got a list of the major differences. We'll just kind of go over them, and then we'll go to them individually. So some of the differences are in structures, the type of APCs, the nature of antigen presentation, the size of the peptide, the responsive T cells, co-receptor responsible, and sources of protein antigens. So we're going to kind of go through these kind of quick. So the first one is the structure. The structure of an MHC class 1 molecule consists of one membrane spanning a chain or a heavy chain produced by MHC genes, and one beta chain or light chain or beta 2 microglobin produced by the beta 2 microglobin gene. In contrast, the MHC class 2 molecule consists of two membrane spanning chains, an alpha and a beta of similar size, and are both produced by MHC genes. Now we're going to look at the types of APCs. A MHC1 glycoproteins are present in all nucleated cells, where in MHC2 glycoproteins are only present on specialized antigens presenting cells, or APCs, including macrophages that engulf foreign particles such as bacteria, dendritic cells that present antigen to T cells, and B cells that produce antibodies. Next, we're going to talk about the nature of antigen presentation. In a MHC class 1, glycoproteins present endogenous antigens that originate from the cytoplasm, where in MHC2 proteins present exogenous antigens that originate extracellularly from the foreign bodies such as bacteria. And we're going to compare the size of the peptides. So in MHC class 1, uh, present 8 to 10 amino acid peptides, and in the MH class 2 presents 14 to 18 amino acid peptides. Looking at number 5, the responsive T cells in a MHC class 1, present antigens to cytotoxic T cell lymphocytes. So you're going to find the TC cells, and in the MHC2, the present antigen to helper T cell lymphocytes, or the CD4 T cells. Number 6 is the co-receptor responsible. In the MHC1, the co-receptor responsible binds with the CD8 co-receptor molecule of the cytotoxic T cell, and for the MHC2, it binds with the CD4 co-receptor molecules on the helper T cells. And the last one on this one we're going to talk about is the sources of protein antigens. In the MHC1, the cytosolic proteins, mostly synthesized in the cell, may enter cytosol through phagosomes, where in MHC2, endosomal or lysosomal proteins mostly internalized from extracellular environment. So we'll go to the last part of the major differences. So the last 4 that we're going to talk about are: number 8 says the enzyme responsible for peptide generation. 9 is the site of peptide loading of MHC. 10 molecules that are involved in transport of peptides and loading of MHC molecules. And then 11, the actual end result. So let's go back to number 8. Number 8 says enzymes responsible for peptide generation. In your MHC1, the enzyme responsible would be the cytosolic proteasome, where in the MHC2, it's going to be the endosomal and lysosomal proteases. For number 9, the site of peptide loading of MHC. In MHC1, it's going to be the ER, or endoplasmic reticulum, where in MHC2, it's going to be the specialized vascular compartment. Number 10, molecules involved in transport of peptides and loading of MHC molecules. For the MHC1, that's going to be the chaperones, the TAPs, and ER. In MHC2, it's going to be chaperones and ER, invariant chains and ER, the Golgi and MHC class 2 compartments, and class 2 vesicles and DM. And then finally, the last difference is the end result. In the MHC1s, presentation of foreign intracellular antigens or altered self-antigens are target cells for destruction, or they target cells for destruction. Now, in the MHC2, the presentation of foreign extracellular antigens induces antibody production and attracts immune cells to areas of infection. So these are the 11 major differences that I found. We talked about how they were alike and went over a brief overview. Here are the references that I used. And Dr. Blair, I'm fine with you uploading this and using it for your library. Thank you.